Assalamualaikum. This is the part 2 for lecture week 10. We're still talking on about immunofluorescence. So this part we're going to focus on the principle behind the procedure of immunofluorescence. There are a few principles for immunoassays, including immunofluorescence. When we talk about immunoassays, that include enzyme immunoassay, ELISA, and also immunofluorescence. The two most commonly being used is the direct and indirect method. Each method have their own advantages and disadvantages. So I hope you can look on the pro and cons between those methods. So first, let's have a look on the direct. Imagine that this is a tissue or cells, human tissue. We want to test for a specific antigen. For example, probably lung tissue that is suspected of being infected by COVID-19 virus. So the antigen is COVID-19 virus on the lung tissue. So we want to detect those COVID-19 antigens. If we are using the direct IF method, staining the antigen on the cell surface with the primary antibody labeled with the fluorophore. So this method is very direct, hence the name direct, and very fast. Straight away use antibodies against the antigen. Second step, we need to wash away the unbound and or any loosely bound antibody, because we don't want for them to produce false positive results here. Washing for a few minutes in PBS with at least 2 buffer exchange will help to eliminate unbound and loosely bound antibody from the sample. And after this, we can straight away go for imaging purposes. For the indirect IF, begins with staining the antigen on the surface with primary antibody. So this primary antibody is antibody being produced to the antigen, specific antibody to the antigen that we are testing or looking for. So this antigen, these antibodies commonly produced by injecting the antigen to animals such as rabbit or sheep, and those animals later will produce specific antibodies against the injected antigen. Their blood will be purified, and the specific antibodies will be extracted. Those specific antibodies from the animal will be used in the IF to detect for the specific antigen. Specific antigen for specific antibodies. Second, we need to wash away the unbound primary antibodies to the antigen. This because we don't want for false positive to occur, because later this unbound primary antibody, they will bound to the specific secondary antibody that carrying the fluorophore. So they need to be washed away to prevent false negative result. The third step is staining the primary antibody with secondary antibody labeled with fluorophore. So this second antibody is specific to the first antibody. So imagine if the unbound primary antibody is still around, so the secondary antibody will stick to them, and later they will be washed away together, which can lead to false negative. As the washing step for eliminating the unbound secondary antibody, because we want to eliminate the possibility of false positive. We need the result to be coming from the area that contain the antigen. So that's why we need to wash away all the remaining unbound secondary antibodies, because we want to detect the signal that supposed to be coming from the area that contain the antigen that we are looking for, not from the antibody that hanging around without specific antigen. So they need to be washed away. And then we proceed with the detection stage, imaging by fluorescent microscope. The stained specimen is placed here, viewed under a UV light with a dark background. So basically, the fluorescent microscope will contain the light source for UV light. The UV light will be shoot to the specimen. If the specimen contain the fluorophore will absorb the UV light, the energy from the UV light, and then they will produce their own color, which can be detected here. So let's have a look on how to interpret the result for IF. This is an example of result. This is a section of brain tissue infected by bacteria. So these are bacterial pathogens, and these are the brain cells. The red dots represent the Bacterium Neisseria meningitidis, the pathogen for meningitis. The antigen is coated by antibody specific to the bacterium with red fluorophore. So that means the specific antibodies is carrying a red fluorophore, which can be detected as red color. As the green area represent the brain endothelial cell. How do we know they are the brain cell? Because earlier we used a specific antibody to brain cell that carrying green fluorophore. So when they are stained, the antibody will stain the brain cells and can be observed as green light under the microscope. As for the blue areas, they represent the cell nuclei because they have been stained with the cell nuclei specific antibody with blue fluorophore. This slide shows the two unique features of IF, which is first, we can locate the location of the specific antigen. We can observe where is the pathogens and where are the cells, even the nucleus. And also shows that IF can be used to detect more than one antigen or antibodies. In this case, in this example, we are detecting 3 different type of antigen in one test. Okay, the advantages of IF includes they can detect against a wide variety of antigens. They are extremely specific and sensitive, so they are very good. They are usually used in the detection of a rare disease and for research purposes. They can pinpoint location of the target, as we have discussed earlier. Also, they can detect multiple antigens or antibodies from the ability to do multiple staining in one sample. And for the disadvantages of IF includes fluorophore undergoes photobleaching as they are exposed to light. That means they are degraded when exposed to light. The need for proper handling of the specimen, as mentioned earlier, the type of specimen for IF includes cells, tissues, and cell cultures. And the needs for specialized, expensive equipment and well-trained personnel. Thank you very much. Have a nice day.